researched Louis Pasteur?
Louis Pasteur is a French biologist, a microbiologist, and a chemist, and is also recognized as the father of biology, microbiology. He's known for his discoveries in the principles of vaccination, how they are used, and is also known for treating milk and wine to stop bacterial contaminations. By 1865, he came up with a remedy which was to treat bacteria in food by heating it. He also showed that germs caused by the puerperal fever in mothers after childbirth in unhygienic situations. He also introduced, this was way before the idea of the immune system or how it was come about or what we know about it today, but he introduced the idea that all infectious diseases could be prevented with prophylactic vaccination and also treated with therapeutic vaccinations, and it will work. It has a high chance of working if applied immediately after an infection.
Okay, so Louis Pasteur was born December 27th, 1822, and he passed away September 28th, 1895. He was born in Dole, France, and he's primarily known as the father of immunology. So he is also known for creating vaccines, disease prevention, pasteurization, and fermentation. He studied at this school, and he got his master's in 1845 and his doctorate in 1847. And here at the school, he was a lab assistant where he studied optical activity. And in 1848, he became a part of the Faculty of Sciences in Strasbourg. And in 1854, he became a part of the faculty in Lille, and this is where he began studying fermentation. So in 1857, he went back to the school he studied at, and he became a director of scientific studies, where he also continued studying fermentation. And in 1865, he began looking into pasteurization. In 1868, he ended up having a stroke, and this delayed him on his studies. So the government ended up building him a new lab, and he was able to go through with creating vaccines. And so the first vaccine he produced was anthrax bacilli, then falciparum, and rabies. And then in 1888, he came up with the Institut Pasteur, which is a biomedical research place. And today they are still creating vaccines and discovering new ones. Okay.
At that time, just like the vaccines he created, the problems were cholera and rabies. But to be specific, it was chicken cholera. And he found out that the bacteria he induced in some chickens was not affecting the chickens. So he would reuse healthy chickens with the same bacteria, and in that whole process, he found out that even with fresh bacteria, he could not still infect the chickens, which means that they somehow got immune to it, because the chickens would not get sick, but they would show symptoms. The next was rabies. The first vaccine he introduced was rabies, and he started growing the virus in rabbits, and then he found out that the rabies vaccine, like the chicken cholera, would not affect the rabbits, but he did that by weakening and drying the affected nerve tissue. So that means when next it was introduced into their body, it wasn't taken up on.
Okay, so he was known as the father of the germ theory, and by this, he found that diseases are caused by microscopic organisms, fermentation. So by this, he studied crystals by the rotation of polarized light in the solution tartaric acid, and when he observed this, they rotated, and then when he looked at paratartaric acid, which is found in wine, he found that there was no rotation when it went through polarized light. And he found that both of these acids had the same composition, and when they were in the solution together, they canceled out the light. For the fermentation process, he found that alcohol in wine was produced by yeast, which is found on the skins of grapes. And under a microscope, he found that the yeast was healthy and budding, but it formed lactic acid, and that the wine then formed into vinegar. And this was amongst when microbes were amongst yeast cells. So he found that bad microorganisms could be destroyed by heating wine between 60 and 100 degrees Celsius. Okay, so to conclude these findings, he researched silkworms. He put healthy silkworms into a nest with ill silkworms and of ones that were suffering diseases, and he found that the environment directly affected the contagion of them. And by this, he found that the spread of disease can be controlled by sterilization, and that studies on yeast microbes could be airborne. And these two studies applied to the transmission of diseases in humans, and this was the start of it. For pasteurization, he found that bacteria removed by boiling and then cooling liquid. And on April 20th, 1862, he completed the first test about it. For cholera, to discover this, he cultured and isolated the microbe that caused diseases in chickens. And he discovered an improved culture medium for the microbe. The method for this was a culture microbe exposed to atmospheric oxygen for prolonged intervals, which were longer than 2 to 3 months, and the vaccine was created and used to immunize animals against lethal challenges of the microbe. The next vaccine is anthrax bacilli, and he found that it killed the bacilli by heating for 10 minutes at 55 degrees Celsius. And on May 5th and May 17th in 1881, he had a bunch of people come to a controlled experiment that he was putting on where he had sheep, 1 goat, and cows, and they were all going to be subjected to the new vaccine. So there was a control group where the animals remained unvaccinated, and then the other ones were vaccinated. And on May 31st, all the animals were inoculated with virulent anthrax bacilli. And then on June 2nd, the crowd came back and they saw that the vaccinated animals were still alive while all the controlled animals had passed away. And then there were 4 unprotected cows that were swollen and feverish. And then this was a start to people realizing that these vaccines could have an impact on their community. For the rabies vaccine, he found that microorganisms causing the disease could not be identified or cultured in vitro, which is in the lab, and he tested them in monkeys and then rabbits, and then they were protected dogs, including ones already bitten by. The rabies shot ended up protecting dogs that were already included. I'm so sorry. Okay, Louis Pasteur found that the vaccine could help protect dogs that had already been bitten by a rabid animal. And July 6, 1885, there was a little boy named Joseph Meister, and he was 9 years old. And he was about to pass away from being bit by a dog. And sorry, let's start fighting again. Okay. So he was close to death, and people wanted him to survive, so they thought that this would be a good chance to test the vaccine. So they tested it on him, and he ended up surviving. So this was the final end to people believing that the vaccines could help people. Okay. Oh, I went back. Okay, and now on to Joshua.
So Joshua Lederberg was a very notable microbiologist of the 21st century, and he worked mainly in bacterial genetics and received the Nobel Prize for it in 1958 at the age of 33, becoming one of the youngest winners ever. The main contribution Lederberg gives to microbiology is the idea that bacteria can sexually reproduce. Before Lederberg's development, it was thought that bacteria could only reproduce by making identical daughter cells. He proposed that bacteria, if able to reproduce sexually, could be useful in research. And as a result of his research and experimentation, he discovered that bacteria did go through genetic recombination and was able to be used in experimentation to discover more about genetics. Another contribution Lederberg made was his discovery of plasmids, which is extrachromosomal material. He also discovered transduction, which is genetic information being passed from one cell to another by a virus. He also sparked interest in finding extraterrestrial life and biological life in space. And so he helped include biological science into NASA's budget after watching Sputnik go into orbit. The last major impact he had towards microbiology was his integration of computers into scientific research.
Okay, so Joshua Lederberg was an American geneticist and microbiologist. He was born in Montclair, New Jersey on May 23, 1925, and he died of pneumonia on February 2nd, 2008. He was brought up in the Washington Heights district of Upper Manhattan, New York, and he received his education in public school 46, junior high school 164, and Stuyvesant High School. From 1941 to 1944, he studied at Columbia College, where he obtained his BA with honors in zoology. From 1944 to 1946, he studied at the College of Physicians and Surgeons of Columbia University Medical School, where he participated in part-time research in the Department of Zoology alongside his professor, F.J. Ryan. He went to the Department of Microbiology and Botany at Yale University, New Haven, Connecticut, as a research fellow of the Jane Coffin Child's Fund for Medical Research, and during 1946 to 1947 as a graduate student with Professor E.L. Tatum. He was awarded his PhD degree in microbiology in 1948, and in 1947 he was appointed assistant professor of genetics at the University of Wisconsin, where he was promoted to associate professor in 1950 and professor in 1954. He organized the Department of Medical Genetics in 1957, of which he was chairman during 1957 to 1958. He received the Nobel Peace Prize in 1958 for his research in genetic structures and function in microorganisms. In 1959, he was entrusted to organize the Department of Genetics at Stanford University Medical School, and later became the professor and executive head of the department. In 1962, he became the director of the Kennedy Laboratories for Molecular Medicine.
While Lederberg was in medical school, he realized that his textbook taught him that bacteria could only reproduce by making identical daughter cells, without any genetic modification. However, Lederberg was not convinced of this. Oswald Avery and René Dubos, some of his contemporaries, said that this idea of identical daughter cells had not been tested thoroughly, so Lederberg decided to find out how exactly bacteria reproduce. In Avery's paper identifying DNA on pneumococcus, discovered that non-infectious causing pneumococcus could become infectious by the non-infectious pneumococcus picking up and integrating virulent DNA from the infection causing pneumococcus. Lederberg decided to look for this same transformation in Neurospora, which is a bread mold.
Joshua Lederberg discovered bacterial recombination and started a new field of research. In 1944, it was the year that Oswald Avery and his group published their paper on the transforming ability of DNA. Lederberg wondered if DNA transformation could be investigated using the genetic tools of Neurospora, so he and Professor F.J. Ryan attempted to do similar experiments using the Neurospora. Unfortunately, they were unable to get the necessary mutants due to the high reverse rates of the nutritional mutant. Lederberg started to think about using a bacterial system, even though there was still debate about whether bacteria had genes or not. An opportunity came to test his ideas when Edward Tatum moved from Stanford to Yale to start a new microbiology lab. Professor Ryan encouraged Lederberg to apply to work in Tatum's lab, and Lederberg was accepted in 1946. Tatum had some E. coli mutants that were suitable for the kind of experiment Lederberg wanted to perform. Instead of trying to replicate DNA transformation into an existing model organism, Lederberg wanted to establish a new system for genetic analysis of bacteria. The current idea was that bacteria did not engage in sex, which made standard genetic analysis impossible. There had been some attempts to detect genetic exchange in bacteria, but the results were inconclusive. Lederberg decided to test this idea. Lederberg chose to use E. coli for the experiments and began the process of mutinization cells and screening them for nutritional requirements. By July 1945, he was ready to attempt to detect sex in cells, in bacteria, rather. Lederberg's plan was to mix bacteria that have different nutritional requirements and select for recombinations with no nutritional requirements. If each strain requires multiple different nutrients, the chance of it spontaneously reverting to the wild type was small. Anything that grows in the absence of nutritional supplements had to be a recombinant. This allowed him to select for individuals that had undergone a genetic exchange. He mixed one mutant that required threonine, leucine, and thiamine with another one that required biotin, phenylalanine, and cysteine. When he inoculated about 1 billion cells on a media lacking those nutrients, around 100 colonies grew. In July, Lederberg presented his observations to the phage group at Cold Spring Harbor. He and Edward Tatum announced their discovery in a brief letter to Nature, later presenting the supporting data in the Journal of Bacteriology. Lederberg had shown that E. coli is sexual, proving that bacteria could be used for genetic analysis.
Okay, so Lederberg and Pasteur both worked with transferring of substances, and Lederberg worked with the transfer of genetic material between bacteria via viruses, and Pasteur worked with silkworms and transferred disease to one another. So that's how their work fits together and how their work impacts science and society today. Institut Pasteur opened in 1888 and continues as one of the premier institutions of biomedical research in the world. Its tradition of discovering and producing vaccines still live on today. And the Academy of Medicine in Paris, which sided with Pasteur, began the process of sterilizing medical equipment. It is still improving the medical sterilization process today. Due to Pasteur's discovery of pasteurization, we are able to preserve and store food for longer periods of time. Vaccines have prevented the spread of disease and contributed to saving lives. Lederberg founded the Department of Genetics in the School of Medicine at Stanford University, and Lederberg also established the Instrumentation Research Laboratory that works to detect life on other planets.
Lederberg also integrated the use of computers and artificial intelligence and brought together some of the top researchers in different fields to further the advancement of science. His findings also discovered transduction, which helped expand genetic engineering and eventually help create gene therapy. He also discovered plasmids, which helped lead to the development of insulin.
And then there's our works cited.
So